All right. Well, we're going to get started with our presentation today. We are going to review the data from our fresh blood versus fixed blood specimen comparison on 25 marker spectral flow cytometry. Just as a refresher, mission at TACO is to make immune measurement reality for clinical trials. And we are going to tackle an important problem that drug developers face on trial. So one of the big challenge for immunotherapy developers collecting specimens for downstream immunophenotyping like flow cytometry is which sample type to pick. So there seem like two bad options. One is to use PBMCs, and about 20% of drug developers do. These can be expensive at 200 to 300 bucks per sample. A lot of times you can miss data like granulocytes, and these have variable viability. The other option is to use live specimens, and this means that you need to have a physical site in every geography, for example, Germany, Japan, Australia, etc. And the problem with this is this can lead to a very high failure rate, 10 to 20% we hear from drug developers. The other option is to try fixed specimens, which is to snapshot the cells in place at the clinical trial site. The problem is fixation hasn't yet been optimized for high dimensional cytometry. So that's what we set out to do with our Toku kit, which is a simple way to fix samples on site. And then the question from drug developers is, does a fixed whole blood sample look similar to a fresh one run on spectral flow? The short answer is yes, and we posted 0.97 correlation between fresh and fixed, but we're going to go through some of the methods beforehand. So in terms of how we set this experiment up, we had 3 donors, and we got whole blood from them and 2 aliquots per donor. And the control arm was fresh, and we'd stain those samples and then put them on a Cytek Aurora. And for the fixed arm, we used our TACO kit called the Toku, where we had 2 steps, stable lyse and stable store fixation, then stained these samples and again ran these on the Cytek Aurora and then compared these head to head. So just as a bit of background on the Toku kit collection process, you start with the blood specimen, you take 2 ml out of that, and you put that into buffer 1. This is a step called prefixation. Then you basically invert that and keep that on hold for about 15 minutes, and then take that entire concoction with the blood in buffer 1 and put that into buffer 2. So buffer 2 is it's a stable store, and then that's the end of the process, and it can be shipped to us on dry ice at -80. So this is a little bit of a snapshot of the collection process we use behind the scenes. And these are the panels we use for fresh and fixed. So if you look here on the left-hand side, this is the fresh, and this is the fixed. And these are pretty much the same except for CD66B, which we included in the fixed samples to exclude neutrophils. And exactly what is it that we compared? We had these fresh samples and fixed samples. We looked by eye at the gating plots, and you're going to see those as well, side by side. And we also compared 36 phenotypic populations and 62 state marker subtypes. So for example, PD1 and CD4. And the measures we looked at were the correlation of population frequencies between fresh and fixed. So let's go through the results briefly. And these are the gating plots from samples stained, live versus fixed. And we're going to start with T cells. So the top over here is live, and at the bottom over here is fixed. So as you go across, you can see gamma delta T cells. These look very similar. Let's go on to CD56 positive T cells. Again, you can see a lot of similarity. CD4 positive and CD8 T cells, Tregs, CD4 positive subsets, and CD8 positive subsets. So looking good so far, and the full gating charts are available as well in our report. Now let's go to B cells. So similarly, we can look at live and fixed and look at total B cells. And again, these look very comparable between the 2 plots. B cell subsets, plasmablasts. So again, we're looking good here in terms of comparability. And we're going to quickly snapshot natural killer cells, dendritic cells, and monocytes. So total natural killer cells, if you look top to bottom, again, these look quite similar. NK cell subsets, total dendritic cells, DC subsets, total monocytes, and monocyte subsets. So yeah, what we're seeing here is really good comparability between these gating plots from live to fixed. So that gives us confidence by eye. What we then want to do is look at the math behind these. So we looked at the phenotypic markers and we looked at the correlation of population frequencies between live and fixed sample processing and found a 0.97 correlation. So the x-axis over here is a percent of top gate level gate for fresh samples. The y-axis is a percent of top level gate for our fixed samples. And you can see here all the cell populations here. So yeah, pretty tight correlation here across all of these different cell types. So now let's look to state markers. So again, very good correlation between state marker positive subset frequencies, like the PD1 and CD4 example I mentioned, between live and fixed sample processing. And you can see over here this correlation plot. So again, the x-axis over here is fresh, the y-axis here is fixed. So in sum, this is the drug developer's dilemma, which is both peripheral blood mononuclear cells and live samples offer a lot of challenges. Fixed samples have been a great alternative, but hard to implement, and the Toku kits can address many of these potential concerns. So this is, again, the drug developer's question. Does a fixed whole blood sample look similar to a fresh one run on spectral flow? And the answer is yes. So we do see a lot of similarities between these, both on the phenotypic level and on the state marker level. What does this enable for drug developers? Well, we think three things. One is a 10 to 20x reduction in failure through centralization of flow. So instead of having a physical site in every geography, having to do inter-site comparisons, you can send to one lab, and you can dramatically reduce your failure rate from the 10 to 20% to sub 1%. Second is you can enable batching of samples, so you can store all the fixed samples until a whole set. So for example, all the time points for a patient is complete, as opposed to being forced to run everything real time. And the third is to reduce your committed processing costs. So if you don't know exactly which samples you want to analyze up front and you want to see how the trial plays out, now you can do that, and that will potentially reduce your costs. So you don't have to make a big financial commitment right at the outset. So yeah, hopefully this gives you a sense of the comparison between fresh and fixed samples. And we can take any questions if there are any on the comparison between the two. And oh, before we get to that, we did have a couple of pre-submitted questions on distinct markers. So let's just go through those. So we had two pre-submitted questions here. So one is on the subject level breakdown of state markers, and then the subject level breakdown of phenotypic markers. So let's just go through those. Okay, so on the subject level breakdown of phenotypic markers, you can see that they had very good comparability here. So if you remember, the top level number was 0.97%. And again, the breakdown by subject here, subject 1, 2, and 3 looked quite good. This is on the state markers. So you can see here breakdown by subject 011, 012, 013. So again, strong correlation between all of these individuals. This is again between fresh and fixed. And then on B cells, the careful eye would have noticed that the B cells were a little bit higher than the other populations. So we do see differences in total B cells. So interestingly, fixed samples are showing a higher percentage of, in this case, of total B cells across all the donors. And so this is an area of active investigation that we're looking at. So yeah, again, very exciting data. I think this enables a whole new class of applications that hadn't been possible before because people had to be relying on either a live or a frozen sample. So with that, I'll pause and see if there are any other follow-up questions. So there's a question of, will we do a comparison of this between spectral cytometry and mass cytometry? Yes, we will be doing, well, close. we will be doing a comparison on fresh versus fixed on mass cytometry, and then we may eventually do a comparison between the two instruments. But probably the next comparison is between fresh and fixed on mass cytometry. So very good question. Next question, is there a specific list of antibody clones you recommend to be able to stain fixed samples as good as fresh samples? So, I mean, it's hard to answer this in general because it requires so optimization. Very good question. So not really is a short answer. We had to do a lot of tuning on our side for the panel. But yeah, we don't necessarily have off the shelf or like generic recommendation. Sorry that we couldn't be of more help, but yeah, here's the actual specific panel we used. So yeah, this is the panel we use. Yeah, in terms of the exact antibodies, that'll really depend on the panel selection. So for Tango, we design panels for customers. Excuse me, we design several standard panels and we optimize those and run those in-house. But yeah, depending on your panel needs, that might vary. So very good question. Okay. Great. So yeah, great that people were able to sign on, and we will post this on YouTube. And if there are any other follow-up questions, feel free to reach out via either email, DM, LinkedIn, etc. So thanks everybody for signing on, and we'll sign off now.